Chemistry in everyday life. Now in this unit you will see about the drugs and different classes of drugs, about the food preservatives and also about the cleansing agents like soaps and detergents. So first let us discuss about drugs. Now what are drugs? Drugs are chemicals of low molecular mass, that is 100 to 500 U. They interact with macromolecular targets and they produce a biological response. Now when I say macromolecular targets, they are the carbohydrates, lipids, proteins and nucleic acids present in our body. So these drugs interact with these molecular targets and they produce a biological response. Now when the biological response is therapeutic and useful, therapeutic means being cured, and useful, these chemicals are called medicines, and we know the medicines are used in diagnosis, prevention and treatment of diseases. Now if medicines are taken in higher doses than recommended, then these medicines in that case will act as poisons. Now we have a term here, that is chemotherapy. Use of chemicals for therapeutic effect is called chemotherapy. Now next, let's see the classification of drugs. Classification. These drugs are classified into 4 types. The first type is classified on the basis of pharmacological effects. Now this classification is based on pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for the treatment of a particular type of problem. For example, analgesics. Analgesics are a class of drug which have pain-killing effect. Now antiseptics. Antiseptics are class of drugs which kill or inhibit the growth of microorganisms. So these are examples for drugs that are classified on pharmacological effects. Now the second classification is on the basis of drug action. Basis of drug action. It's based on the action of drug on a particular biochemical process. For example, we know antihistamines. They are class of drugs which inhibit the action of the compound histamine, which causes inflammation in the body. Okay, so these are a particular class of drug is used in the treatment of a particular problem. So there is a second classification that is based on the drug action. Now the next is on the basis of chemical structure. It's based on the chemical structure of the drug. Drugs classified in this manner, they share a common structural feature and they often have similar pharmacological activity. For example, sulfonamides. Sulfonamides have a common structure. Okay, so this is the classification that's based on the chemical structure of drug. Now the last classification is based on the molecular targets. Molecular targets. I told you drugs, they interact with biomolecules, or the molecular targets are carbohydrates, lipids, proteins and nucleic acids, and they are called the target molecules or drug targets. And drugs possessing some common structural features may have the same mechanism of action on targets. So this classification, that is classification on the basis of molecular target, is the most useful classification for medicinal chemists. So these are the 4 classification of drugs. Now next let us see about the drug target interactions. Now, we know that these macromolecules like proteins, nucleic acids, lipids, carbohydrates, they perform various functions in our body. We know the function of protein. Proteins, we know the function of protein, which are example enzymes, and enzymes are proteins which perform the role of biological catalyst in the body. And receptors, that's another type of protein which are crucial to communication system in the body. Now there is another class of proteins called carrier proteins. They carry polar molecules across the cell membrane. And nucleic acids, they have coded genetic information for the cell. Lipids and carbohydrates are structural parts of cell membrane. So let's study about the drug target interaction, taking enzymes and receptors as examples. So we know enzymes are proteins which perform the role of catalyst, biological catalyst in the body. And receptors, they are crucial for communication system in our body. So first let's study the catalytic action of enzymes. Catalytic action of enzymes. Now, there are 2 major functions which enzymes possess when they act as a catalyst. And the 2 functions are, the first function of an enzyme is to hold the substrate for a chemical reaction. Now when I say substrate, here with regard to this medicines, we can say substrate means it can be a virus, a bacteria or allergens. So the function of enzyme is to hold the substrate for a chemical reaction. Now on the enzyme, suppose this is our enzyme structure. There are active sites on this enzymes. Active sites. So active sites of this enzyme, they hold the substrate molecule in a suitable position so that it can be attacked by the reagent effectively. Now substrates, they bind to the active site. So I can show a substrate having the same shape like this. Okay, so this is our substrate, and I told you substrate means virus, bacteria, etc. They will go and attack this active sites on the enzyme, and they attack the active site through a variety of interactions. Or what are the different forces that hold the substrate on the active site of enzyme? That is ionic bonding, hydrogen bonding, van der Waals interaction or dipole-dipole interaction. So these are the various forces that hold the substrate molecule on the active site of enzyme. Now, so that is the first function of enzyme, that is to hold the substrate on its active site. Now the second function of enzyme is to provide the functional group that will attack the substrate and carry out the chemical reaction. So these enzymes will provide the functional group necessary for the chemical reaction to take place between the substrate and enzyme. Now next let us see about the drug enzyme interaction. Drug enzyme interaction. So now we have our enzyme, and in the active site of enzyme we have a substrate molecule present. When I say substrate, a virus or a bacteria. Okay, now what will happen when we take medicine? So we are going to discuss about drug enzyme interaction. So we have a virus, or we can say a substrate in the active site of enzymes. Now drugs, when taken, they inhibit any of the above mentioned activities of the enzyme, that is, the two activities or two functions of the enzyme is to hold the substrate and second is to provide the functional group. These can block the binding site of enzyme and prevent the binding of substrate or can inhibit the catalytic activity of the enzyme. Such drugs are called enzyme inhibitors. So, what are enzyme inhibitors? They are drugs which block the binding site of enzyme and prevent the binding of substrate or which inhibit the catalytic activity of enzyme. Now, drugs inhibit the attachment of substrate on active site of enzymes in two different ways. So, these drugs will inhibit or prevent the attack of substrate on the active site of enzyme in two different ways. So, let us see the two different ways. The first method is drugs compete with natural substrate for their attachment on the active site of enzyme. Such drugs are called competitive inhibitors. So, we have a term called competitive inhibitors. Now, suppose this is our enzyme. We have the active site here. Now, we have a substrate and we have a drug. Now, what do you mean by competitive inhibitors? That is, the drugs compete with the natural substrate. So, this is our substrate and this is our drug. So, this drug will compete with this natural substrate to get attached on the active site of the enzyme, and such drugs are called competitive inhibitors. So, what will happen when drug is taken? They will replace the substrate, they will compete with substrate and this drug will come and occupy the position of active site of the enzyme. Now, the second method is the second method of drug-enzyme interaction is some drugs, they do not bind to the enzyme's active site. They bind to a different site of an enzyme which is called allosteric site. So, this is our enzyme. Now, the drugs, they will not come and attack the active site. Instead, what they do, they will come and attack a site which is called allosteric site. So, I am going to show the allosteric site. This is our enzyme and here this is the allosteric site. So, allosteric site means some drugs do not bind to the active site of the enzyme. Instead, they bind to a different site of an enzyme called allosteric site. So, what will happen? This drug will come and attack this position. Okay. Now, this binding of inhibitor at allosteric site changes the shape of active site in such a way that the substrate cannot recognize it. So, when this drug come and attack this allosteric site, what will happen to the shape of this? Shape will change of the active site. It changes like this. So, what will happen? The incoming substrate will not identify this active site of the enzyme. Okay, and what happens? They cannot attack. So, these are the two ways by which the drug enzyme interaction take place. The first method is the drugs will compete with the natural substrate for their attack on the active site of enzyme, and such drugs are called competitive inhibitors. The second method is the drugs will go and bind to the allosteric site, which in turn changes the shape of the active site of the enzyme so that the substrate cannot identify the active site. Now, if the bond formed. Now a bond is formed between this drug and the enzyme. It cannot be, if a strong bond is formed, then it cannot be broken easily, then the enzyme is blocked permanently, and the body then degrades the enzyme inhibitor complex and synthesizes a new enzyme. So this is how drug enzyme interaction take place. Now let us see receptors as drug targets. I told you we are going to study two examples for drug target interaction. One is drug enzyme that is interaction, second is drug receptor interaction. So next we are going to see drug receptor interaction or receptors as drug targets. Okay, and you know the function of receptor, it helps in the communication system in our body. Now receptors are proteins that are crucial to both bodies' communication process, and majority of these are embedded in cell membrane. Now receptor proteins are embedded in cell membrane in such a way that their small part possessing active sites projects out of the surface of the membrane and opens on the outside region of the cell membrane. So these receptors also has got active sites. Now in the body, message between two neurons and between neuron to muscles is communicated through certain chemicals called chemical messengers. And these chemical messengers are received at the binding sites of receptor proteins. So I told you there are active sites for the receptor protein. So these chemical messengers, they are received at these active sites of the receptors. To accommodate a receptor, the shape of the receptor site changes. This brings about the transfer of message into the cell. This chemical messenger gives messages to the cell without entering the cell. So if we draw the diagram, we can say, we can show it like this. Suppose this is the receptor receiving a chemical messenger. Chemical messenger is coming. This is the chemical messenger. They will bind to the active site of the receptor, and when it comes to attack the active site, the shape of the messenger changes. So the shape of messenger changes, and after the communication is done, what will happen? This chemical messenger will leave that binding site and it regains, that receptor cell regains its original shape. So there is receptors as drug targets. Okay, now there are large number of different receptors in the body that interact with different chemical messengers. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure and amino acid composition. Now we have two terms to study, that is antagonist and agonist. Antagonist and agonist. Now, what are antagonists? Antagonists are drugs that bind to the receptor site and inhibits its natural function, and they are useful when blocking of message is required. Now, agonists are types of drugs that mimic the natural messenger by switching on the receptor. They are useful when there is lack of natural chemical messenger. So these are the two types of drugs, that is antagonist and agonist. So with this we complete the classification of drugs and about the drug target interaction. Thank you.